The BioClock Studio Winter 2020 presents the Circadian Transcriptomics Dry Lab Tutorial, Part 404, Visualizations. In this tutorial, we will discuss how to visualize RNA sequencing data. To understand how sequencing data are obtained, please see the second video in this series. Communicating results is a critical part of research. Accurate and clear visualizations are the primary means of relaying the results of a study. In this video, we will discuss common methods for reporting many types of omics data: scatterplots, PCA plots, heat maps, and volcano plots. A scatterplot allows easy visualization of the expression levels of specific genes under two different conditions. An example of this would be to compare expression of time-restricted versus ad libitum fed mice. Time-restricted feeding is plotted as TRF on the x-axis, and ad libitum is plotted as ad lib on the y-axis. For each gene, the transcripts per million, or TPM, is plotted. The higher the TPM of a gene, the more highly that gene is expressed in the sample. With this plot, it is easy to see which genes are differentially expressed between conditions. Genes that lie on the 45-degree line are expressed equally in both samples. Genes that lie above the 45-degree line have greater expression in mice fed ad libitum, or genes that lie below the 45-degree line have greater expression in the TRF mice. As you can see in the scatter plot, gene X has a TPM of 11.5 in TRF mice and a TPM of 10 in ad libitum mice. Thus, gene X is more highly expressed in TRF mice. The same type of representation can be made for levels of proteins or metabolites in proteomics or metabolomics experiments, respectively. While scatterplots help researchers visualize differences in individual genes across samples, PCA plots, which stands for Principal Component Analysis plots, compare biological samples by entire sets of genes. The goal of PCA analysis is to determine which variable in an experiment is affecting data points more than another variable in the study. In this case, she wanted to know if the feeding regimen or mouse genotype is most responsible for changes in gene expression observed in a time-restricted feeding experiment. Let's go over how one of these plots is constructed. We begin PCA analysis by identifying the sources of variation in gene expression between individual samples in the experiment. To do this, we first draw a scatter plot comparing the normalized expression of each gene in a sample against its expression in every other sample in the experiment. In our example, you can see a scatter plot comparing the expression of genes in sample 1 on the x-axis plotted against their expression in sample 2 on the y-axis. We next draw a trend line summarizing the spread in XY values in our dataset, shown in our plot as the red and blue lines. As you can see, the red line is longer than the blue line and summarizes the expression of genes that tend to have similar expression levels in sample 1 and sample 2. The blue line summarizes variation in gene expression that differs between the two samples and is much shorter than the red line, showing that the expression levels of most genes are correlated between the samples. For the purpose of PCA analysis, we call the red line Principal Component 1 and the blue line Principal Component 2, or PC1 and PC2. For each gene in the data set, we can calculate its contribution to the variation along each of these lines and can see that genes at the edges of PC1 determine the range of the data and length of the line and therefore are considered to have a high influence on PC1, since they have a greater influence on the total amount of variation than those at the center. As this analysis is iterated across all samples in the data set, additional trend lines or principal components are generated summarizing all of the directions of variation between samples. These can be plotted in a scree plot summarizing the magnitude of each principal component. We select the two principal components with the largest extent of spread or length of trend lines summarizing variation between samples to move forward with the analysis. To understand how the collection of genes in each sample describes that sample relative to all other samples in the experiment, we then calculate the PC1 and PC2 scores for individual samples using a mathematical formula that takes into account the expression levels and influence on PC1 and PC2 for every measured gene. This gives us a table with overall influence scores for PC1 and PC2 for every sample in our experiment as shown here. To draw the PCA plot, we make a scatter plot using the PC1 scores for every sample on the x-axis and PC2 scores on the y-axis. If groups of samples show gene expression patterns that are more similar to each other, such as biological replicates or similar genetic or environmental conditions, they will cluster together in this plot. In this case, genotype was shown to influence the data more than feeding regimen. For other datasets, a PCA plot may show that samples at a particular time point from several genotypes are more alike than other time points from a single genotype. In summary, this plot shows how similar or different individual biological samples are to each other on the basis of large sets of genes, or metabolites, or proteins, that are changed in similar directions within samples. The previous two visualizations are helpful for elucidating differences or similarities in gene expression between different groups, but what if we want to visualize the rhythmicity and peak phases of genes within the same group, as many circadian biologists do? We can use a heat map to accomplish this goal. A heat map is a way of visually representing values in a matrix. In the heat map shown, each row corresponds to a gene, each column corresponds to a sample, and the color is an indication of the read count for a specific gene in a sample, reflecting increased expression, red, and reduced expression, blue. Notice how the samples are arranged by the hour at which the sample was collected in a 24-hour day. Arranging the data in this fashion makes a heat map useful for visualizing the rhythmicity of genes because rhythmic genes will show easily detectable cyclic patterns. Creating a heat map is as simple as entering your RNASeq count matrix into a heat map constructing program. However, it is common for researchers to normalize or transform the RNASeq data before entering the data to get the best representation. This means that the expression of a given gene is compared to its own expression at different time points, rather than an absolute expression. Without this step, samples of genes of low absolute number of reads, such as gene A, would all appear blue, or genes of high absolute number of reads, such as gene D, would all appear red, preventing their relative rhythmic change from being detected. Researchers instead can normalize the data by computing and plotting the z-score, or maximum standard deviation from the mean, for each gene. This normalizes the gene to itself and isolates the coloring to each row, so you can visualize the relative changes to expression level of each gene. As you can see from the heat map on the right, this reveals the rhythmicity in gene D and emphasizes the rhythmicity of gene C. An additional step of importance is arranging the genes on the y-axis by peak time to reveal patterns. As you can see, we have shuffled the positions of gene A and gene C so that rhythmic genes with a later peak time are located lower on the heat map. This arrangement has the effect of creating the distinct color gradient associated with circadian heat maps that aids in the visualization of circadian phasing. Applying the same principles of normalizing and arranging by peak time with an increased number of samples and genes results in a heat map similar to the ones you might see while reading a circadian research paper. For example, we can use the same approach to construct a heat map from this larger set of rhythmic genes sampled for 24 hours at a frequency of 2 hours. As you can see, the genes are ordered on the y-axis by descending peak time, resulting in the distinct batting pattern alluded to earlier. Genes that display similar peak phases may then be selected and subjected to further downstream analysis like gene ontology, since they might be involved in coordinating a time-of-day specific biological process. The final plot we will cover in this tutorial is one that is constructed with the output of a DE-seq2 analysis, described in Dry Lab Tutorial Part 3. Volcano plots incorporate the p-values and fold change values generated from that differential expression analysis and help us to visualize which genes are most relevant from a biological and statistical standpoint. Looking at this volcano plot, each point represents the change in expression between two conditions for a unique gene. As indicated in the legend, the red points are genes that are upregulated in one sample relative to another, whereas the blue points are genes that are downregulated based on the magnitude of their fold changes and statistical significance. Notice that the x-axis is the log base 2 of the fold change. The generally accepted threshold for fold change is 2. This is because gene expression fold changes of less than 2 are often assumed to be too small to be of biological significance. The log base 2 of 2 is 1. Genes beyond 1 and -1 on the x-axis have a fold change greater than 2, which is why only genes beyond these lines are highlighted with color. But also notice that not all points greater than 1 or less than -1 on the x-axis are colored. This is because changes in gene expression must also be statistically significant. Notice that the y-axis is the negative log base 10 of the p-value. To be statistically significant, the p-value must be below 0.05. The negative log base 10 of 0.05 is 1.3, so genes located above 1.3 on the y-axis have p-values less than 0.05, indicating a statistically significant difference. The genes in color meet both the fold change and p-value thresholds. These are the genes that are considered most relevant, and additional studies would focus on these genes. Together, the tools and methods we have discussed in this tutorial series allow scientists to understand changes that occur in an omics sample because of a genetic or environment perturbation and bridge the gap between these changes in an observed phenotype. By sampling every protein-coding RNA expressed in the liver in a time series over greater than a 24-hour period, it is possible to discover the sets of genes that change their steady state or circadian expression patterns in response to clock gene mutations and/or time-restricted feeding. Gene ontology analysis can then curate the repertoire of biological and physiological processes that are known to be regulated by the genes found in the RNAseq analysis and point to groups of genes that might be responsible for the observed phenotypes. Lastly, if little is known about the resulting gene set, investigators can generate new cell or animal models to study these genes or pursue them as possible drug targets to improve outcomes in disease.